Alright, the next unit we're going to start looking at doing is on Gregor Mendel and his genetics. Gregor Mendel, who he was a modern day genetics, kind of actually known as the father of genetics from the mid 1800s. He was an Austrian monk, and during that time, like many monks that have quite a bit of time on their hands, he dabbled in some gardening, and he noticed one of the things that he was working with were peas. And he just done certain qualitative analysis, started to take a look at it and saw that certain appearances were different than others, even though they are all peas. And so he started to use some experimental analysis and try to then utilize quantitative analysis, look at what he can do in numerical amounts to collect data based upon certain traits that were being expressed. Now, this was an excellent example of the scientific methods. He had that first questioning technique. So what he was looking at was if he could manipulate, he could cross-pollinate. Now, remember, as sophomores, we talked about how some plants they can self-fertilate, but also cross-pollination with the help of the abiotic factors of the wind. In fact, here, Mendel played the abiotic factor as the wind, and he would take a particular flower and cross it with another flower of a different color. So cross-pollinated between true breeding parents. And now we're going to say in terms of true breeding parents, we're going to call it our P generation, our parental generation. Now, that P generation, if that's on the top, could have kids. Those kids are going to be known as the F1, or the first filial generation. Now, if those kids have kids, those are going to be known as the F2 generation. Okay, so F1 is the kids of the parents, F2 are the kids of the kids. Now, some of the things that Mendel actually looked at, some of the traits that he looked at, and we're going to use a particular term, a vocab term for traits. And that physical appearance trait, we're going to call it a phenotype. A phenotype. Now, here's an example of some of the phenotypes that are going to be used. So I'm just going to write that term out here for you. Phenotype. The phenotypes are the physical traits. And so those physical traits that we're looking at again are the flower color, whether the flowers are white or purple, the seed color, whether they're green or yellow, maybe the shape. Are they round seeds or wrinkled seeds? Pod color, green or yellow, if they're inflated or if they're pinched. Axial versus terminal flowers, where are they going to be located? And if they're just tall or short plants. So these are all different examples of phenotypes that he would actually look at. Now, in doing so, what he ended up finding out is that he was crossing true breeding plants. And what this means by true breeding is that every time a purple flower was mixed with another purple flower, they always got purple. White with white always got white. So those are what we're going to be calling true breeding, or homozygous. So the term true breeding is also meant as homozygous. Now, what he has found out is that when he crossed them, he wanted to identify, even though if they're homozygous, maybe one might be more dominant over the other. And so in doing so, he found out that if he crossed a purple with a white, 100% of the time were purple. 100% of the time. So if going back to sophomores, you know that purple then means it is what in comparison to the white? Should be homozygous dominant. Again, white is also homozygous. It is just not the dominant trait. It is a recessive trait. So purple is a homozygous dominant. White was the homozygous recessive. So if we're looking at particular traits, let me insert a slide real quick. Let's see. Let's just do this. Let me insert a quick slide. So if we remember back to our karyotype, which we talked about just briefly in our last chapter, we had all of this little charts. Remember, we had one chromosome was mom's, one's was dad's. What we're looking at are particular traits that are actually going to be found on those chromosomes. And those traits are what we call alleles. And being that they're at a specific location, we call that the loci. The location that those traits are actually located at. Now, if I was to zoom in just on those two there, we could say that this one was a big A. Let's actually keep it pertinent to what we were talking about. Let's erase the A. Let's make this a big P for purple and a little p for white. Okay, that would be an example of what we found in our generation here. Big P, little p, because they inherit the little one from mom or dad, whoever was the white one, and the big one from that one. Okay, now if they were to cross, they were to go ahead and cross then two of those individuals. Let's zoom out of this. So if we had this crossed with another one, we could figure out a particular ratio we might have. Now this brings in our mathematical box that we've seen before, which we call a Punnett square, where we have our big P and our little p represented, big P and little p. So if I do my mathematical box, I notice that there's only one box that has two little p's in it, so only a 1 out of 4 chance to get white. But the other three that are here, or three out of 4 is purple. Okay? So, in terms of what we're talking about here again, what did Mendel's findings actually mean? Traits come in alternate versions. So, purple versus white flower, these traits are again the alleles, and this is what I just showed you on that last slide. The alleles for purple flowers. And then they're at a particular location. That particular location is the loci. Now remember in our last chapter we talked about homologous pairs. This one was mom's, this one was dad's, and in the S part of interphase we would have a identical one here, an identical one here, held together somewhere in by the centromere. Okay, we're not going to be worrying about that now since we just finished through the whole process, so we're going to get rid of that. But remember, that's where those things are coming from. So now if we just look at the two individual chromosomes, one's from mom, one's from dad's, they have it on a particular loci or a location of the gene. This is singular now. Locus is the specific spot of the gene, is where we're going to find that set of nucleotides that's going to code for that allele, and that allele being that particular trait that we want to see expressed. Now, traits are inherited as discrete units for each characteristic. An organism inherits two alleles, one from mom, one from dad. That's why we are considered diploid, having our full set of chromosomes. The other ones, which we talked about in meiosis, how we end up getting our haploid set. What they end up meaning, you might even find that some are masking others, which we talk about dominance versus recessive. If it is a dominant trait, it's going to be fully expressed. If it's a recessive trait, it is still there. That trait is still there, it's just masked, meaning that you have some sort of over-dominant trait overshadowing the other, even though it is there. Now, it's a difference between how an organism looks, is the genotype versus the phenotype. We already mentioned the term phenotype already in here. The genotype we have not written out, but that all that refers to is the genetic makeup. So if I wanted to show you again our genetic makeup of our example we had before, we could have our two big P's is purple. Our big P, little p is still purple, but our little p is white. Okay, these three orientations are the genetic or genotype, where these three are the physical appearance, the phenotype. Okay. Now, when we end using representative letters, flower colors, the alleles, capital P or lowercase p, true breeding versus true breeding. Remember, they both have the same. In this sense, they are both dominant, though are both lowercase. They have the same genotype letters. That is considered homozygous. If, let's do this. If they look like this, they are considered heterozygous because it has one of each, one big, one little. So going back to our example, again, we have our true breeding homozygous dominant purple plant crossing with our true breeding homozygous recessive plant. 100% of the time we get a phenotype of purple, but all of them, if you take a look at them, all of them, in terms of their genetic orientation, is heterozygous for their genotype. So then if this is the F1 of the hybrids. Now if I were to cross those again, I may ask you what percentage of my F2 offspring are going to be homozygous for particular traits. I could look down at it. If I wanted to figure out how many of them, what they'd end up being, 25% white, what my genotype would be, little p. These two here would be big P, little p, but I'd also have one big P, big P. That's when I'm doing my 3 to 1, 3 of them are purple, one of them are white. Because all three of these have at least one big P to mask. Okay, and they're showing what our illustration would look like. Okay, homozygous, heterozygous, remember same alleles versus different. You guys kind of get that point. Genotype versus phenotype. Two organisms can have the same phenotype but have different genotypes. So what that's telling you is that they may still look the same, being dominant in homozygous, so we can't really tell if it's dominant or heterozygous. So if I want to do a test cross to identify if it's homozygous or if it's homozygous dominant, homozygous recessive, I want to test it with something that's homozygous recessive because I know the only way that I can have a recessive trait expressed, meaning actually visually see, is if both traits, both alleles are recessive. So, dominant phenotypes, it is not possible to determine the genotype of an organism with dominant phenotype by looking at it. I can't tell it's purple, that's great, but I can't tell if its genotype is homozygous dominant for it or heterozygous for it. So this is when I do that test cross. I cross it with one that I know is recessive, and being that I know it is recessive, if I have any offspring in the F1 that show up as being white, then that tells me that I have a heterozygous individual. However, if they all show up purple, that tells me that this purple flower was homozygous dominant for the trait. So there you go. I have 100% purple or 50% white and purple. Now, take a quick peek at some of the laws of heredity, laws of segregations. When gametes, we just talked about how gametes, sex cells, egg and sperm, are produced during meiosis. These homologous chromosomes, one from mom, one from dad, separate from each other. So to take a look at those, how they separate themselves out, each allele for the trait is packaged into a particular separate gamete. So we end up with 4 cells at the end of meiosis. All the alleles can be different. And in terms, remember, even crossing over had occurred, which creates even some more of that genetic diversity. So what you get in terms of interdependent assortment and segregation is a huge variety. Okay, let's go back to that. There we go. Okay, so I'm taking a look at what meiotic event creates this. Remember that when we go through interphase, we get our dual set. Meiosis 1, the diversity, the alleles are starting to be separated, and eventually you start to see in meiosis 2, you get your whole individual ones separated out. But remember, meiosis 1 is where we are starting to see the variation take place. Okay. Looks like we're wrapping up on our time for this first section. We'll call that quits there, and then we'll start off here for part 2 of the Mendel video.